How does stomach cancer metastasize? Gastric adenocarcinoma. Imagine a tiny cancer cell in the lining of the stomach that somehow manages to spread far beyond its original spot. That's what metastasis is all about. In gastric adenocarcinoma, the most common type of stomach cancer, this process is quite complex and involves several steps. First, cancer cells break through the basement membrane of the stomach lining and invade nearby tissues. This is often driven by a process called epithelial to mesenchymal transition, or EMT, where cancer cells change from being stationary to becoming more mobile and invasive. During EMT, cells activate pathways like TGF-beta, WNT/beta-catenin, RAS, and Notch, which help them degrade surrounding structures using enzymes called matrix metalloproteinases. These enzymes cut through extracellular matrix proteins, clearing a path for the cancer cells to move. Once the cells have invaded local tissues, they can spread through the lymphatic system. The stomach is rich in lymphatic vessels, making it easy for cancer cells to enter these channels and travel to nearby lymph nodes. This is often the first step in metastasis, and cancer cells can settle there, forming secondary tumors. Genes such as ERB2, CLDN11, and CDK12 can promote this spread by helping cancer cells stick to and migrate through lymphatic vessels. Cancer cells can also enter blood vessels, a process known as hematogenous spread. Once inside the bloodstream, they travel to distant organs like the liver, lungs, or even the brain. To do this successfully, they must survive in circulation, exit the blood vessels, and then grow in new tissues. Mutations in genes like ERB2 and activation of pathways such as MAPK and EGFR support this process. Another common route for stomach cancer to spread is within the abdominal cavity, called peritoneal dissemination. Cancer cells shed into the peritoneal fluid and float freely, resisting a form of cell death called anoecus. These cells then attach to the lining of the abdominal cavity, invade the underlying tissue, and form new metastatic nodules. Enzymes like matrix metalloproteinases help break down tissue barriers here as well. The fatty areas of the peritoneum, such as the omentum, provide a nurturing environment that encourages tumor growth, aided by nearby fat cells called adipocytes. Throughout this process, several molecular and cellular factors come into play. Signaling pathways like TGF-beta, WNT/beta-catenin, and Notch promote EMT and cell mobility. Proteolytic enzymes like matrix metalloproteinases help cancer cells invade tissues. Cancer cells also develop resistance to cell death, especially when they detach from their original site. The tumor microenvironment, including interactions with stromal cells, immune cells, and fat cells, influences how well the cancer can spread and establish itself elsewhere. Genetic changes, such as overexpression of HER2 or mutations in other genes, further enhance the metastatic potential. In summary, gastric adenocarcinoma spreads through a coordinated series of steps involving local tissue invasion, lymphatic and blood vessel dissemination, and peritoneal spread. Each step is supported by complex molecular signals and interactions with the surrounding environment. Understanding these mechanisms helps researchers develop targeted treatments and offers hope for better management of stomach cancer. If you or someone you know is dealing with this disease, always consult healthcare professionals for personalized advice and care.